Our final podcast in our series on cytogenetics is going to deal with chromosomal translocations. Abnormal chromosome structure can result from translocations that are either unbalanced or balanced. Unbalanced would be rearrangement that causes gain or loss of chromosomal material. This can produce serious consequences to individuals or to offspring. On the other hand, balanced rearrangements do not cause gain or loss of chromosomal material. Those would usually have minor health consequences. And let's assume that these are occurring in gametes for our discussion. So what is a translocation? A translocation is the transfer of one part of a chromosome to another part of the same or a different chromosome. It results in rearrangement of genes. If we think about translocations occurring on the same chromosome, that's a good thing in a sense that that's what homologous recombination does during meiosis. If the translocation is occurring between different chromosomes, that you might think of could be not so good a thing because that might lead to genetic consequences. Most translocations occur on the autosomes. Before we talk about those, let's talk about sex chromosomes and pseudoautosomal regions. The pseudoautosomal regions are regions on the X and Y chromosome. There's 2 or 3 regions that share homology with each other and undergo very high levels of recombination. And recombination is actually absolutely important for chromosomes to pair so that they can segregate properly during meiosis 1. So the X and Y chromosome have to pair during meiosis 1 so they can segregate. Now there's a pseudoautosomal region 1 that's at the distal end of the X and Y chromosome on the short arms. There's an obligatory crossover point in a 2.6 megabase region that's required for proper segregation of X and Y. Now it turns out that the SRY gene is just proximal to the pseudoautosomal region in the Y chromosome, and unequal crossing over in this pseudoautosomal region between X and Y can lead to XX males and XY females. This is just a hypothetical example of what could occur, so it's showing crossing over leading to an XX male or an XY female. The pseudoautosomal region acts like an autosome where there's exchanges on the XP region with X and Y. The SRY gene, as you may remember, is the sex-determining region on the Y chromosome. It's expressed during embryonic development. It encodes a product that interacts with other genes to initiate development of an undifferentiated embryo into a male. This is the example showing the balanced chromosome rearrangement between the homologous chromosomal regions. The pseudoautosomal regions of X and Y cross over in meiosis. The SRY region contains the testis-determining factor, and it normally resides on the short arm of the Y chromosome. The gene product, as we said, determines if the embryo is male. Translocation of the SRY gene, as we said, can lead to XX males or XY females or even to hermaphrodites. So here showing normal crossing over between X and Y with the SRY region maintained on the Y chromosome. Here showing crossing over that would occur below the SRY region, so that gets translocated onto the X and you'd have XX male and then an XY female. And this is rather interesting. This type of crossover where it occurs below the SRY gene may occur in 1 in 20,000 live births. That might suggest that there may be something like 15,000 individuals in the United States that have this unusual translocation. There may be up to 3 babies a year that are born in Tennessee with this translocation. The pseudoautosomal region 1 on X and Y chromosome also contain genes called the SHOX genes. The SHOX genes stand for short stature homeobox genes. So there's the SHOX genes on the X chromosome, and there's the SHOX Y gene on the Y chromosome. And this just shows their chromosomal locus. You need to have two functional copies of the SHOX genes for normal height. So I might ask, how would this relate to short stature in Turner syndrome? Turner, you have 45X, so you only have one copy of the SHOX gene. You're missing the other SHOX or the SHOX-Y gene. There's also a condition called Langer-mesomelic dysplasia and Leary-Veal syndrome that result from SHOX mutations. These behave like mutations on autosomes. Genes located on the pseudoautosomal 2 regions of the X and Y chromosome are necessary for vesicle fusion, and they code for proteins related to interleukin receptors. There is no known function for genes that are on the pseudoautosomal 3 regions of the X and Y chromosome. Rearrangement or translocation between homologous chromosomes and the autosomes generally may have no genetic consequences. You might ask, why is that the case? It's because you're exchanging material on the same homologous chromosomes. But what would happen if there's a rearrangement between non-homologous chromosomes? Here's an example that shows a reciprocal balanced translocation between chromosome 6 and chromosome 3. So these are derivative chromosomes. Here's a derivative chromosome 3 with a little bit of chromosome 6 on it, and a derivative chromosome 6 with a little bit of chromosome 3 on it. The offspring here, this child receives a derivative chromosome 3 and a normal chromosome 6 from the carrier parent, and normal chromosomes from the other parent. And we'll come back to this. Reciprocal translocations are caused by breaks on non-homologous chromosomes, so there'll be exchange of genetic material. Carriers of balanced reciprocal translocations usually have normal phenotypes, but the offspring may have a partial trisomy or a partial monosomy and an abnormal phenotype. So the previous example showed a partial trisomy for chromosome 6. So here you see the partial trisomy for chromosome 6. You should also see that this child is a partial monosomy for chromosome 3, because here is the derivative chromosome 3. It shows this chromosome 3, but it's missing the other region of chromosome 3, so it's a partial monosomy for that region of chromosome 3. Reciprocal translocations are usually balanced, and they don't carry a risk for disease to a carrier unless one of the breakpoints interrupts gene function, and that actually may be maybe a 5% chance that can occur. Reciprocal translocations can cause problems in meiosis because of segregation in meiosis 1 may lead to gametes with unbalanced chromosome complements and genetic disease in an individual. Here's a normal individual. Here's an individual with a reciprocal balanced translocation. When these pair during meiosis, you can see that you may get unusual segregation. So here is an example of normal segregation, whereas this chromosome segregates with that chromosome. Here is a balanced segregation, where this one segregates with that one. But then you can see the unbalanced ones, where this will segregate with that, or this one will segregate with that one. Another series of unbalanced segregations. This one segregates with this one, or this one segregates with that one. Here's an example of a Robertsonian translocation, and these occur when the long arms of acrocentric chromosomes fuse at the centromere. So in humans, Robertsonian translocations can only occur between chromosomes 13, 14, 15, 21, and 22. In the case shown here, the long arms of chromosome 13 and 14 have fused. There's usually no genetic consequences because the individual has the full information on chromosomes 13 and 14, maybe has lost a little bit of the short arms. This individual, the karyotype would be designated 2n equals 45, translocation 13, 14. The individual with this balanced translocation doesn't have any genetic consequences because they may have lost a little bit of the ribosomal genes, but remember the ribosomal genes are repetitive. Robertsonian translocations are responsible for a small percentage of Down syndrome phenotypes, and we'll ask you to think about that. Here is the translocation between chromosome 21 and chromosome 14, for example. This individual is 2n equals 45. Translocation 2114 has a normal phenotype, but the offspring could have problems as a result of pairing at meiosis 1. This shows the individuals with Robertsonian translocations. They have normal phenotypes since no genetic material is lost, but since they have only 45 chromosomes, pairing and segregation at meiosis 1 can lead to potential problems. This shows possible chromosome combinations and gametes that are going to be transmitted to offspring. Here you can see this one will segregate with that one normally like that, or you may have that one segregating. This could segregate normally like that, or that one can segregate. This one can segregate normally with that, or that one can segregate. So you look at those gametes being fertilized with gametes from a normal parent, and you can see the types of combinations that you'll get. Possible combinations of gametes in the offspring of normal parents and a translocated carrier parent. And I'm going to bring this to the next slide just to make it a little bit easier to see. So here would be the gametes in a normal and translocated parents with the possible combination of offspring. These individuals, you'll have a monosomy, a trisomy, a monosomy, and in these individuals, normal, a balanced translocation, and a translocated Down syndrome. These individuals, the monosomic 21, the trisomic 14, and the monosomic 14, don't survive to term. Now you might expect equal frequencies of normal individuals, balanced translocation individuals, and individuals with Down syndrome. But it turns out that if mothers have this translocation, 10% of the births will be Down syndrome. If fathers have the translocation, 2% of the births will be with Down syndrome. That's a lot higher than 1 in 800, which is the typical frequency of babies born with Down syndrome. Abnormal chromosomes may come about from deletions. You may have microscopically observable deletions, so you may have a terminal break. In this case, a hypothetical sequence, if you have a terminal break here, you'll have a deletion like that. You may have an interstitial deletion, so you may have a break here and a break here will result in something with a deletion like that. Kruder-Short syndrome is caused by a deletion in the short arm of chromosome 5. Williams syndrome is caused by a deletion in the long arm of chromosome 7. So you might represent the karyotype 46XY with a deletion in the long arm. Various microdeletion syndromes can be recognized with high-resolution chromosome banding and fluorescence in situ hybridization. I'm not going to ask you to memorize these for an exam. They're just here to give you some examples. More chromosomal abnormalities, you can get duplications where there's unequal crossing over or offspring of reciprocal translocations. Usually the consequences are not severe. Inversions, there may be two breaks and reinsertion of a fragment at the original site, but in an inverted order. So you can have pericentric inversions, which include the centromere. Paracentric inversions do not include the centromere. Inversions usually do not produce disease in a carrier, but there may be problems in meiosis and disease in the offspring. And you can have ring chromosomes. Here's an example of a pericentric inversion where the inversion involves the centromere region. Here's an example of a paracentric inversion where the inversion is just on the long arm of the chromosome. Here's an example of a ring chromosome that's formed where the two little pieces are lost and the remaining chromosome forms a ring. Chromosomal anomalies can relate to developmental delay and intellectual disability. There may be alterations in facial morphogenesis. There can be growth delays, short stature, poor weight gain. There can be congenital malformations, especially heart defects, because many genes can be affected. And finally, chromosome rearrangements in relation to somatic cells can cause cancers. For example, chronic myelogenous leukemia is a reciprocal translocation between chromosome 9 and 22, and that's shown here. The derivative chromosome 22 is called the Philadelphia chromosome. There is a case where the Abel proto-oncogene is moved to the long arm of chromosome 22. Reciprocal translocations between chromosome 8 and 14 can cause Burkitt lymphoma. A deletion in the long arm of chromosome 13, for example, can cause retinoblastoma. Here's the example of the reciprocal translocation between chromosome 9 and 22 causing chronic myelogenous leukemia. Here's an example of that condition with fluorescence in situ hybridization, and here is showing the Philadelphia chromosome with the fused Abel oncogene product. Basically, part of the breakpoint cluster region from chromosome 22 is fused with part of the Abelson oncogene on chromosome 9. That fused BCR Abel oncogene product codes for constitutively active tyrosine kinase. That activates many cell cycle proteins, leads to uncontrolled cell division, may inhibit DNA repair and apoptosis. Additional mutations occur like that, and that will tip the balance. Some of these additional mutations may be in the RAS genes and in the p53 tumor suppressor gene.